Thank you, and thank you to MLA for inviting me to speak today. So we've heard a lot about my Irish colleagues and what a great job they've done, but I'll start out on a positive note and say that Australian geneticists are none too shabby either, and just show you a few world firsts that have been achieved in the past 18 months as a place to start. So this is Australian genomics highlights. And one of these really is genomic breeding values for eating quality traits in sheep. I don't think any other country has genomic breeding values for intramuscular fat and shear force in sheep. This was developed by the Sheep CRC, and it's a little bit visionary what they're trying to do here, because as you well know, you don't get paid for eating quality of lamb, but it's anticipating a future where if you select too hard on just lean meat yield, you're going to compromise meat quality. So it's about giving breeders the tools to balance up eating quality and lean meat yield now, so we don't run into that problem in the future. Just another example, DataGene, which does the dairy cattle evaluations in this country. In December, I think it was, they released the heat tolerance breeding value for Holstein and Jersey cattle. And this is a great example of how to put together really big data sets, managing big data. This was a result of merging weather station data and herd recording data, millions of records collected over 10 years or so, to come up with how tolerant dairy cattle are to heat, how compromised their milk production will be if there's a heat wave, for example. And now dairy cattle farmers have the tools to select the bulls that are going to be more heat tolerant, to give them resilience to those heat stress events now and into the future. So that's a really nice example. Another example of a world first is the single-step breeding values that are there for Brahman cattle for days to calving. And this is really a fantastic tool to improve fertility in this breed. And that was released last year, quite a few months ago now. So it's also intended to give you a snapshot of where we are now. We have genomic breeding values. They're out there for a number of traits. But this talk is really about where do we go next? What are the next steps? And what are the new technologies that we can bring to bear? So what I'm going to try and cover is exactly that. Where do we go next? So a big part of that is making our genomic breeding values more accurate. The more accurate they are, the better job you can do of selecting young bulls with really good genetic merit, the faster you go in terms of genetic gain. Something else we've observed in the dairy industry as well is that the more accurate the genomic breeding values are, the more confidence people have to use them. You get greater rates of adoption, and it just goes up exponentially. Okay, we want to get away, as Tom's just been saying very nicely, we want to get away from working just within breed. We want to have genomic breeding values that just work. So by that I mean they work regardless of breed, crossbreed or composite. They work in any type of cattle. And they work for traits that we've had difficulty in selecting for in the past, like fertility and feed efficiency. So genomic breeding values for growth, there's not a whole lot of point in that. It's pretty easy to select for growth. But for traits like fertility, they're much harder traits to select for. So genomics can really play a role there in balancing up the gains we get for growth and fertility. And then finally, we get onto the really fun bit about new technologies. And I think those new technologies take us into a different space where the cost of genomic testing gets cheap enough then we can start to think about using them as a management tool and not just for genetic improvement. So let's tackle those points one by one. More accuracy and faster gains. How do we get there? So I'm going to take a leaf out of the dairy industry book here and just take you through what they did to get to very accurate genomic breeding values. And this was work that's been done over the past 4 or 5 years. So what they did was say, we need these very large reference populations to drive the accurate genomic breeding values. So we need lots and lots of cows with really good data and that are also genotyped to give us our accurate genomic breeding values. In dairy, it's really lovely industry to work in because what you can do is go into the national database and you can see which dairy farmers are doing a really good job of recording data. It's just there in the database. And you can say, let's just go in and genotype those cows. And then we'll add them in to the system and we can calculate the genomic breeding values from that really good data plus the genotypes. So that's pretty much exactly what we did. We mined the database. We found about 100 herds around the country that were doing an exceptional job of recording not just milk production, but also fertility data. And we said to those herds, do you want to work with us in this GInfo project to develop these accurate genomic breeding values? Across those about 100 herds, there were 30,000 cows. So that starts to give you an idea of the scale at which you need to operate to get these really accurate genomic breeding values. So 100 herds, 30,000 cows, all with good fertility and milk records. They were genotyped, and that was the basis for the genomic breeding values that are now coming out of DataGene. And just to give you an idea of the accuracies that were being achieved, the accuracy is how well the DNA test or the genomic breeding value is predicting the true genetic merit of the animal. So if it's 0.2, it's reasonably average. If it's up around 0.9, it's exceptional. So after doing those 30,000 cows, production we could predict with an accuracy of 87%. So that means for a young bull, you can take a DNA sample, run it through the system, and you get a prediction of the accuracy of his genetic merit for production that is not 100%, but not far off. It's pretty good. Traits like fertility are harder to crack. The genetic component is lower, the heritability is lower, so you end up with a genomic test that has an accuracy about 65%. But that's still pretty good. You can still make very good selection decisions on that. Feed efficiency was a bit interesting. We actually had to put together the reference population for feed efficiency, because for feed efficiency, you have to measure individual intakes of animals, and you can only do that on research stations. So that's why the accuracy is lower there. Just the numbers of animals recorded are a bit lower. But those numbers, those accuracies, are high enough that the dairy industry could make this fundamental shift from selling bulls based on progeny test results to selling young bulls based on a DNA test alone. The accuracy of the information was high enough that dairy farmers had confidence to use those bulls. And in fact now, greater than 50% of the semen straws sold in the dairy industry are from young bulls, 18 months, 2 years of age, that have genomic breeding values attached to them. And that's a real shift from, I guess, 3 years ago, when the majority of bulls would have been progeny test bulls that were producing semen for dairy farmers to use. Okay, so that's a big shift. Another question is, does it work? The promise is using these genomic breeding values is going to increase our rates of genetic gain. And in the dairy industry, the promise was you could reduce down the generation interval, so the age of the bulls that you were using, far enough that the rates of gain would actually double. So has that really happened? Does it work? And there was a very nice paper came out a couple of years ago now. It's actually from the US industry, where they've done something very similar, but showed that yes, in fact, the rates of gain were doubling or even greater. And the graph that you see up on the screen is not for milk production, it's actually for fertility rate, daughter pregnancy rate. And what it shows is when genomics came in the US dairy industry, which was about 2008, 2009, the rate of gain for daughter pregnancy rate really started to kick up. Before that, a couple of decades before that, it was actually declining, started to turn around a little bit, and then genomics has allowed it to really accelerate. Particularly if you look at the green line there, that's the genetic values for the young bulls that are really at the pointy end of the industry, contributing a lot of the genetic material that's the foundation for the next generations. And that's for a trait that's been really quite hard to improve in the dairy industry. And it's brought into much better balance, gains for production and gains for fertility. Okay, so that's the dairy industry. Beef industry, more complicated. More breeds. We don't have data coming in every single day from our animals. How do we make gains? One thing is we do want these genomic breeding values that work across breeds, across composites, across crossbreds, and we want them for traits like fertility. So how do we get these multi-breed genomic breeding values that are accurate? And again, it's all about putting together these large reference populations where you have animals recorded for the trait that you want the genomic breeding values for, and you also have them genotyped for the markers. So there's two approaches you can take here. One is you can take a structured approach, and for multi-breed, one thing you need to have is different breeds running under exactly the same environmental conditions, so in the same cohort. So you can take a structured approach to that, run it on research stations. The other approach is to use commercial data. And in this country, for Northern Australia, both approaches are actually happening at the moment. So to take you through that, the structured approach is exemplified by the Repronomics project run by David Johnston. And what this project does is use industry sires, so leading industry sires of Brahman, Droughtmaster, and Santa Gertrudis breeds, and uses these sires to breed heifers on 3 research stations. So that's Bryan Pastures, Spyglass and Douglas Daly in the Northern Territory. And on these research stations, you can have these cohorts of heifers of the different breeds running head to head to enable these multi-breed genomic predictions. And all these animals are, of course, genotyped, so they form a reference population. And there's some great data coming in from this that's already contributing to accuracy of genomic breeding values. The other approach is to use commercial data, and that's exemplified by the Northern Genomics project, I guess, which I head up. And so far in this project, we're working with 33 herds. You can see the map of where the herds are. It's all in Queensland at the moment. We'd like to shift across the north of the country. We've got plans to do that, but at the moment we're in Queensland. But even within Queensland, of course, there's a huge range of different environments, and we've tried to cover all those in our selection of herds. And the key criteria for being in this project are that you have large cohorts of heifers and ideally multiple breeds or crossbreeds in those cohorts of heifers. So then you get this comparison of the breeds and crossbreds in exactly the same environment. The repro data we're looking at, because it's on such a large scale, we can't do the intense measurements, the very detailed measurements that something like Repronomics is doing, but we are measuring have heifers cycled or not by 600 days as a measure of age at puberty, and also do they get pregnant on the first rebreed, so postpartum anestrus. There's also traits like temperament, fly scores, tick scores, udder scores in there as well. The aim is to get 30,000 cows across the north, so this is quite a large-scale project. Another point about this project is geneticists are very good about dismissing the environment completely and just treating it like a nuisance. In this project, we're actually trying to get some good information on the environment, so we're working with the pasture monitoring people to get pasture growth on these properties, and also fecal NIR to get pasture quality. So that'll give us a much finer resolution on these environments. Okay, so that's just two of the projects contributing to multi-breed genomic breeding values that are well underway. The next question is, will it work? And we've got a little bit of data we can play with here to answer this question. And this particular data that I'm going to show you comes out of the Smart Futures project, which was led by Brian Burns. So in this project, there were 7 collaborating herds, and there was about 3,900 heifers of either Brahman, Santa Gertrudis, or Drought Master breed. They were scanned for presence of, or absence of a corpus luteum at 600 days. So were they cycling or not? Similar trait to what we're collecting in the Northern Genomics project. And this is an indicator of age at puberty. And all these heifers were genotyped for 24,000 SNPs. And the question we looked at in this data was, does the information from the different breeds help you with your target breed? So if you've got Droughties, does using information from the Santas and the Brahmans help you get more accurate genomic breeding values? So to show you the answer, this graph just shows you how accurately we can predict a whole herd. So this is quite a stringent validation. This is dropping out each herd in turn, predicting what their heifers should have been doing just from the DNA markers. So we get a genomic breeding value for each heifer in the herd, and then we correlate it with what they actually did. So you get a genomic breeding value, were they cycling or not, just from the DNA, and then you correlate it with, yes, they were actually cycling from the scanned result, or they weren't cycling. So that's what you see on the y-axis, is that correlation between what you predicted from the DNA and what they actually did. And we've dropped out, we've predicted each herd in turn. And what you can see, or what the graph shows, is what you get for that correlation when you just do it with inbreed and when you put all the breed information together. And it says when you do it with inbreed, when you use 24,000 markers, it's not particularly good. Your correlations are pretty low. It's pretty lowly accurate genomic breeding values. We used 24,000 SNPs to do this. We also had very high density SNPs, so 800,000 DNA markers across the genome. When we did that, when we used those SNPs, and that was coming down from their sires actually, the accuracies went up a little bit. But the real gains were when you combined all the breed information together. So to predict the Droughties, you didn't just use the droughty information, you also used the Santas and the Brahmins all together. So in a style of multi-breed evaluation. And you can see for Santa Gertrudis it works really well, Drought Masters is working well, and even for the Brahmins you're getting an increase in the accuracy there. So it's just a suggestion that this multi-breed approach is going to work pretty well, and all the breeds involved are going to benefit from it. Okay, so that's multi-breed, and projects are underway to develop that. To move a couple of steps into the future, when we're going to have real success, I think, is when genomics is used not just for breeding, but also for management. If you think about the scale of the two different uses of genomics, if you're talking about just for breeding, you're limited to all the breeding herds, all the stud herds and so on around the country. If you talk about genomics for management, all of a sudden you're talking about millions of cattle. You're on a completely different scale of genotyping altogether. And when you go up those orders of magnitude in scale, you can start to really drive down the price of the testing. When you operate it in orders of millions instead of tens of thousands, you really do start to have some pricing power. So this would be a good place to get to. So I'm going to talk about new technologies here. You could actually use the current technologies for, say, feedlot screening, but I'm going to talk about some of the new technologies that are coming along that might result in big drops in price. Okay, so one of my favourite potential new technologies that we could translate into the cattle and sheep industries is this idea of very rapid sequencing on basically a USB stick. So what that guy's holding in his hand, that's actually a genome sequencer. So you can put a drop of blood or a drop of tissue in there, and it will read off your genome. It will go through and potentially sequence all 3 billion ACTs and Gs of your genome. And in the bottom picture there, you can see this thing just plugs into a laptop. So you drop the bit of blood in, run the thing, and you get your genome readout. And that's pretty phenomenal. I have to tell you at the moment it takes half an hour, but it's reducing in time. The time to get your genome is reducing all the time. This technology is already being applied in the field. A colleague of mine, Lachlan Coyne, Professor Lachlan Coyne, he's using it in hospitals to screen patients when they come in to see if they have a bacterial disease, and specifically, do they have bacteria that's going to be resistant to normal antibiotics. So these very hard to treat bacterial cases. So you can imagine the patient comes in, bit of blood from the patient, drop it into the thing, sequence the bacteria in the blood, and then work out which drugs to treat the patient with. So that's actually happening, and he's in my building there at University of Queensland. The other place the technology is being applied is in Africa, and this is in the breeding of cassava varieties. Don't know if you've ever tasted cassava, but it's like a big tasteless potato. It's a pretty ordinary thing to eat, but it is a supply of energy, and it's a real food security crop. But it's a clonal crop, which means every single cassava plant is genetically identical, and it does, because of that, it's very susceptible to being wiped out by viruses. So a researcher from Western Australia, she's actually using this bit of equipment, this USB stick sequencer, to go out into the field, take a sample from cassava plants, and work out what strains of viruses they have on the spot. So this happens in the field. She can tell the farmers, look, you're about to be wiped out, or don't worry, it's not a particularly virulent virus that's there. So if it can be used in Africa, in cassava fields, surely we can take this bit of technology and develop something that I've termed crush side genotyping, which is, just imagine your cattle are coming down the race. You take a tail hair or an ear punch at one end, put it into this machine, and by the time they hit the head bale, you can make a decision. So the genome's been processed, you've got all your genomic breeding values and so on, and you can decide that animal's actually a cull. If it's a heifer, she's never going to make it. If it's a steer, doesn't have the genetics to marble well or whatever, so it doesn't go to the feedlot, goes to live export or something like that. So that's the potential here. And we've got projects just starting at the very early stages in this area. Yep, it's good. So if we're talking about feedlot performance, and I've just mentioned it a couple of times, genetics is definitely important for things like potential to marble, but for traits like feed efficiency, days on feed to reach market specs, there's other genomes that the cattle carry that are very important. And that is, of course, the rumen microbiome. So if you think of your cattle, they're carrying around this enormous vat of microbial soup that has all these different bacteria, different organisms that allow them to digest the feed and turn that into high quality beef or milk if it's a dairy cattle and so on. That same bit of technology I just showed you, it can also be pointed to sequencing the rumen microbiome, all those bugs in the rumen. Just like genetics, there's a lot of variation between animals in what genes they carry. They also carry different abundances of these different bugs that do this job of digesting the food. Some have rumen microbiome profiles or a bug complement that makes them more efficient than other animals. So if you want to predict feedlot performance, ideally what you do is get the genome of the animal plus the rumen microbiome of the animal to make a prediction of how well it's going to perform. Okay, so you could actually add the two things together to get a more accurate indication of feedlot performance. Now, when I was down in Victoria, we had a very small crack at this. So using a very small number of animals, we actually had a crack to see, could we predict from the rumen microbiome plus the genome how feed efficient animals would actually be. And it's a very small study. The stats that come out they're not significant and so on, but it just shows you the potential. So there were 28 heifers in this study. They all had individual feed intakes measured, so we knew their feed efficiency. We also had their genotypes, so we had genomic predictions for feed efficiency, and we also had rumen microbiome profiles. So we knew what bugs they carried and the relative abundance of the bugs. So the question is, if we put those two things together, the genomic breeding value for feed efficiency plus the rumen microbiome profile, can we do a better job of predicting efficiency? And as I said, this is a very small study, but just to give you a feel for what's possible, what you see in the table there is the correlation between what we predicted and how the heifers actually performed when they were on feed and we knew their individual intakes. So just from the rumen microbiome, from the profile of the bugs, relative abundance and so on, we could predict their feed efficiency with an accuracy of about 0.5. It was actually reasonably good. The genomic prediction, its accuracy was about 0.3 to predict how well a heifer's actually performed. When we put the two things together, you came up with an accuracy of 0.57. So you got an additional benefit from knowing both the rumen microbiome and the genome of the animal as well, and combining that. So, very small study, but just shows you what might be possible in the future. So to finish up then, there is big projects running to put together these reference populations to improve the accuracy of genomic breeding values for high traits like fertility. And this is going to lead to more balanced breeding. It's going to enable you to more easily balance up selection for growth and for fertility and make gains in both traits. Multi-breed genomic breeding values, certainly not here yet, but a lot of the work underpinning these multi-breed genomic breeding values, at least in Northern Australia, has begun. Repronomics will be a big contributor to this, and the Northern Genomics project has just got started as well. And then we can predict any breed, any composite, any cross. There's exciting technologies coming that are basically here. We just have to do a good job of translating them to livestock that bring up the potential for things like crush side genotyping, profiling the rumen microbiome, as well as doing the cow's own genome to get accurate predictions of feedlot performance, for example. And just finally, thank you to all the people involved in the Smart Futures project, Northern Genomics project, and down in Victoria that ran the study that I just showed you. So thank you.